A-Level Biology 9700, Paper 4, Variant 2, October, November 2024, Question 10. This is the last question of this question paper. So blood glucose concentration is maintained around a set point by homeostasis. Explain the principles of homeostasis. Before going into detail, I think we should discuss what is the homeostasis and what is a set point by homeostasis. So homeostasis is a process by which the living organisms are able to maintain a stable internal environment despite the changes in the external surroundings. So during this process of homeostasis, the body has a set point for different variables. As for example, for those variables can be the set point. Number one, it can be temperature. And our body has set point 37 degrees centigrade. On the other hand, the second example can be the blood glucose concentration, which is 80 to 100 milliliter per deciliter. So it can be 80 to 100 is a set point, you can see. So in this way, uh, body has a set point for different variables. So I have given example of this. After this set points, you can see there are receptors. Receptors. Receptors, these are the sensors or the sense organs which receive the or which detect the change due to a stimulus. And what can be the change to a set point? That is, if there is increase in the environmental temperature, then our body temperature will also change. It can be, as for example, the outsider temperature is 40 degrees centigrade or 50 degrees centigrade or 45 degrees centigrade. So our body temperature will also raise. But and also but our body temperature remains the same, that is 37 degrees centigrade. In any condition which is outside, in any temperature which is outside, because of this, because 37 degrees centigrade is a set point. So on the other hand, the blood glucose concentration is also, there can be a change in the blood glucose concentration. If we are taking high sugar diet, then what will happen? The body glucose concentration will be increased. As for example, it can be more than 130 or 140 or 150 milliliter per deciliter. So it can be raised. So it will be detected by our body receptors, which they will detect the change which is taking place in these different types of variables. Then the message will be sent towards the control center, the force control center. So control center will receive information from the receptors and process it. It compares the detected level of the variable to the set point and determines the appropriate response needed to restore balance. Balance is compulsory, so the brain, particularly the hypothalamus, often act as a crucial control center. So control center, hypothalamus, which is control center, temperature receptor, they will detect the change and then message will be sent to the control center, and from control center will process it, and then there will be the message will be sent towards the effectors. Effectors are the, you can say, the muscles of the body. So these can be the organs, tissues, or cells that carry out the commands issued by the control center. So these actions work to counteract the initial change and bring the variable back towards the set point. For example, the body temperature has been changed to 37 to 40 degrees centigrade. So there will be sweating, lot of sweating. There will be some other changes which will take place inside the body, and temperature will come back to its set point, that is 37 degrees centigrade, by a process which is called as negative feedback. Because it is coming back from the condition which was changed due to a stimulus, that is from 45 degrees centigrade or 40 degrees centigrade to a set point, that is 37 degrees centigrade. So this is called as mechanism of negative feedback. And you can see that if we are taking high sugar light, then what will happen? The concentration of sugar will be increased in the body from a set point. So what will happen in that case? The receptor cells, they will be detected the change in the blood glucose concentration, and then what will happen? Then the uh, activation of the uh, pancreas will take place, and as a result of activation of pancreas, it will start secreting the insulin. It will start secreting the insulin, and that insulin will take up the glucose from the cells and from the muscles, and then what will happen? The extra glucose will be stored into the liver in the form of glycogen. In the form of glycogen. So in this way, the insulin secretion is decreased and it returns to its set point, that is around 80 to 100 degree, 100 milliliter per deciliter. So I have written this answer as well because we have to explain the principle of homeostasis. So when a change occurs away from a set point, already explained what is set point, a maintained condition in the body. So the control center triggers a response that opposes or reverse that change, bringing the variable back to the desired range. For example, for normal functioning of the body, it is very compulsory, that is, the different variables should always be at the set point. So reverse that change, bringing the variables back to the desired range. For example, if blood glucose level rise too high after a meal, the pancreas releases insulin, which promotes the set uptake of glucose by cells, thus lowering blood glucose back to the set point. Once the glucose level returns to normal, insulin secretion decreases. So this is the whole sequence of the different events which take place when body controls the or when the body try to keep the homeostasis inside the body in case of any change in the variables. Now, next question is glycogen. Already told you in this question, that is the extra glucose is stored in the liver in the form of glycogen. So glycogen phosphorylase catalyzes the conversion of glycogen to glucose in liver cells. So glycogen phosphorylase is that enzyme which breaks down the glycogen to glucose in liver cells. In that case, when there is less concentration of blood sugar due to fasting or due to any reason, so in that case, the glycogen uh, is broken down into glucose to make the level of glucose up to a set point. So the production of glycogen phosphorylase is coded by the gene PYGL gene. This is a specific gene. A mutation, a sudden change in PYGL in the structure or number of the DNA. So in PYGL leads to a condition called glycogen storage disease type 6, in which glycogen is not broken down efficiently. This is compulsory. This is the normal functioning of the body. That is, the glycogen should be broken down into glucose in that case when there is less sugar level inside in the blood. So suggest and explain why cell signaling by glucagon is likely to be affected in the liver cells of a person with GSD6. So glycogen binds and initiates signaling in the liver cells of a person with GSD6. The deficiency in glycogen phosphorylase means that the intended outcome of this signaling, the rapid release of glucose from glycogen store, is severely compromised. This leads to problems in maintaining blood glucose homeostasis, particularly during fasting. And students, that when our body is not properly function means many issues can be generated inside the body. So as in the question, it is clear that this enzyme, which is glycogen phosphorylase enzyme, glycogen phosphorylase, helps to break down the glycogen into glucose. So here, cell signaling by glucagon. Glucagon is that uh, hormone which is secreted instead of insulin. In that case, when there is less glucose level inside the blood, so which normally it stimulates the breakdown of glycogen into glucose. So glucagon's normal function is this: in only it is secreted when there is by the pancreas, when there is less blood sugar concentration to a set point, that is less than the set point. So to make it up to a set point, in that case the glucagon is secreted, and that glucagon activates the glycogen phosphorylase enzyme. So this activated glycogen phosphorylase then catalyzes the breakdown of stored glycogen into glucose, so which is released into the bloodstream to raise blood glucose level. But in individuals with GSD6, the deficiency is functional glycogen phosphorylate, mean that even if the glucagon binds to its receptor and initiates the normal signaling pathway, the key downstream enzyme responsible for glucagon breakdown is not sufficiently active. In that case, it will not active, and then what will happen? Then the glycogen will not properly broken down into glucose level, and it will not fulfill the needs of the body. So in this, what will happen in this case? So it will this due to this less activation of this enzyme, the properly it will not break down and body will suffer. So here in this way, I would like to say the glucagon signaling might still have maybe accumulation of glycogen in the liver. Glucagon breakdown is impaired. Individuals with GSD6 often experience hepatomegaly due to excessive enlarged liver, you can say. They due to the excessive accumulation of glycogen in liver cells. That is what happens with the persons who have this problem, that is the uh, inactive form of glycogen phosphorylase, due to which they have enlarged liver due to accumulation of glycogen, because glycogen is not properly broken down. So in that case, the enlarged liver will be there, and the body will not properly functioning. Glucagon can, at that time, you can see the glucagon can still bind to its receptors and initiate signaling in the liver cells of a person with GSD6. The deficiency in glycogen phosphorylase means that the outcome of this signaling, the rapid release of glucose from glucagon stores, is severely compromised. This leads to problem in maintaining body glucose homeostasis, particularly during fasting. And during fasting, the body will suffer a severe decrease in the glucose level, which uh, can, and in that case, the body has to face the severe consequences. That is the due to less glucose level, the body has to suffer a lot. So in that case, the body is not properly functioning if the this mutation in PYGL leads to this glycogen storage disease. You can say the one major problem can occur, that is the liver will be enlarged due to accumulation of this glycogen. Extra glycogen will be there and it is not properly breaking down. Now the next question is glycogen synthesis uh, catalyzes the conversion of glucose to glycogen in liver cells. The production of glycogen synthesis is coded for the gene GYS2. So GYS2 gene, a mutation in the GYS2 leads to condition called glycogen storage disease type 0, in which glycogen is not formed efficiently. So suggest what the consequences would be if a person with GSD0 has a meal rich in glucose. So in glycogen storage disease type 0, a mutation in the GYS2 gene leads to a deficiency in liver glycogen synthesis. Liver glycogen synthesis. Synthesis. So this is that enzyme which is essential for the conversion of glucose to glycogen. This enzyme converts the glucose into glycogen. In case when there is high blood sugar level. So normally after a glucose rich meal, blood glucose levels reach insulin is released, which stimulates liver cells to take up glucose from the bloodstream and convert into the glycogen for storage. However, in GST0, the deficiency glycogen synthesis, the liver cannot efficiently convert this excess glucose into glycogen. As a result, the blood glucose, so this glucose will not change into glycogen. It will not be stored. And then what will happen? The storage mechanism is severely impaired. This liver will not be able to replenish its glycogen reserves even after a high glucose intake. So what will happen with impaired glycogen synthesis, the body will rely more on other pathways to deal with the excess glucose. And this could include, you can say, the increased conversion of glucose to fatty acids. So this glucose can be changed by the body into fatty acids instead of changing them into glycogen. And then it will, if it is changing into fatty acids, so what will happen, that the in this way there will be high levels of lipids in the body over time. So the second is this glucose will be secreted into the urine. More of the glucose will be secreted in the urine, and because you can say if blood glucose level becomes very high, it can overwhelm the kidney's ability to reabsorb the glucose, leading to glucose in the urine. So glucose is an osmotically active substance, so its presence in the urine can draw more water. You know, if more glucose is in the urine, it will be concentrated, due to which more water will be drawn out from kidney instead of reabsorption. Which will lead to dehydration. So when blood glucose starts to fall, the liver can break down glycogen to release glucose into the bloodstream. So in this way, I would like to say that the body will lead to dehydration, and due to dehydration, the problems can occur into the body, and which can be the severe. So I have written this answer as well. If a person with GS consuming a meal rich in glucose will experience a significant and prolonged rise in blood glucose levels due to liver's inability to store glucose and glycogen. So in that case, the liver, as don't have glycogen synthase enzyme, which helps to change the glucose into glycogen. So due to lack of that enzyme, there will be the liver will be unable to change the glucose into glycogen, and glycogen and glucose will not be stored. And due to less storage of of this glucose, it will become into the urine, or it will be changed into others products, that is the fatty acids, which will increase the fatty acids in you can say the lipids concentration in the body. And secondly, the more glucose will be secreted into the urine, and due to more concentrated urine, due to this glucose level, the more water will be draw out from the kidneys, and due to more the body will leads to the more water draw excretion instead of reabsorption. The more water will be excreted, and due to more excretion of a of this, what will happen? The body will have to face the or to experience a significant and prolonged problems in the body due to this. So this can lead to hyperglycemia and an increased reliance on alternative and potentially less efficient pathways for glucose disposal. Furthermore, it highlights the paradoxical situation in PSTO, where individuals can have high blood glucose after eating but are still susceptible to hyperglycemia during fasting due to the lack of liver glycogen stores. So body has to face severe problems due to this inabilities. So it was all about question number 10. Thanks for watching. Please like, share, and subscribe my channel. If you have any problem, you can send a message to me. Thanks.